Thanks for the introduction and thanks for inviting me. I'm going to be talking about how we can use machine learning in phylogenetics and population genetics. So today I'm going to tell you about three different projects. I'm going to start by telling you about some work from my postdoc on trying to infer phylogenetic trees with machine learning approaches. And then I'll talk about some stuff that my lab is working on now with demographic model selection and machine learning. And then finally, I'm going to talk about model misspecification and domain adaptation. Okay, so to start with, how can we infer phylogenetic trees using machine learning approaches? There are several challenges that arise when we try to infer a species tree from a multiple sequence alignment. These include things like heterotachy, where we have variation in rates of evolution, incomplete lineage sorting, where gene trees might disagree with the species trees and each other, gene duplication and loss, and introgression. All of these things add heterogeneity to our data and make inference more challenging. And using model-based approaches that can accommodate all of these different processes is not always tractable. And so we know that heterogeneity complicates phylogenetic inference, and we also know that machine learning can be an excellent tool for trying to accommodate heterogeneity. I'm going to be talking about supervised machine learning approaches today. And in supervised machine learning, our goal ultimately is to make some prediction. So for example, in phylogenetics, we might want to predict a tree topology. To do that, we have to have what are called training data. Training data are data sets where we have data and we know the history or label for that data set. So for example, if our goal was to predict whether or not there was introgression between species, we would need a lot of genomic data from species where we knew whether or not there was introgression. Of course, that's not very feasible in phylogenetics and related fields. So instead, we typically use simulations under the model or models that we're interested in to generate these training data. So to use machine learning in phylogenetics and population genetics, we have to rely on these simulations to generate the training data, and then we can extract information or features from that data and use some algorithm to build a model, which can then be used to make predictions on new data like empirical data. This approach has proven really successful in fields like population genetics. There have been a lot of papers about how powerful these approaches are for doing things like inferring parameters in population genetic models. These approaches have also been applied in phylogenetics, although the uptake has been a little bit slower here. Here are a couple of the early papers doing this, and these approaches found that machine learning worked really well for inferring phylogenetic trees. However, both of these approaches focused on inferring quartet trees or trees for taxa. To understand why that is, we just have to go back to how we actually use supervised machine learning in phylogenetics. Before we can make any predictions about new data, we have to train this algorithm. And to train this, we have to have simulations under the relevant models. In the case of a 4-taxon tree, that's not too hard. We just have 3 different models because there are 3 different potential tree topologies. And so it's fairly straightforward to generate data under these models and build a machine learning algorithm that can take as input a multiple sequence alignment and predict probabilities for each of these 3 trees. So in this case, we just have to perform simulation under 3 different tree topologies. However, this quickly gets a lot less feasible. So with as few as 5 taxa, we already have 15 unrated trees in phylogenetics. And as we add taxa, this is going to get worse and worse. So with as few as 10 taxa, we have 2 million different trees that we need to consider. Since in phylogenetics, the model space is the tree space at a minimum, the number of possible trees is going to become prohibitively large, even with moderate numbers of taxa. It's not feasible to try and simulate thousands of data sets under each of 2 million different trees to train a machine learning algorithm. Luckily, this problem isn't unique to phylogenetics, isn't unique to machine learning approaches in phylogenetics. And typically when we want to infer trees in phylogenetics, using, for example, maximum likelihood, we might use heuristics to explore tree space. And all this means is that we use approaches that don't force us to look at every tree in the tree space. So for example, in this 5-taxon tree space, we could propose a random tree topology, and then we could look at trees that were similar to that tree, and we can compare them using some criteria like the likelihood. We can accept trees that improve the likelihood, and we can continue to do this until we move through tree space, hopefully to the best tree shown here in green. And this is an approach that's used in many of our tree inference algorithms. So we wanted to explore whether we could use a heuristic approach to infer topologies with machine learning. And for this, we turn to generative adversarial networks, or GANs. A GAN is a network that includes a generator and a discriminator. The generator is going to create fake data. So in this example, the generator is creating fake images of handwritten digits. And the discriminator is going to try to distinguish the fake data from real data. At each iteration, the discriminator will try to get better at distinguishing fake and real data, and the generator will try to get better at making realistic-looking fake data. And hopefully, by the end of training, we have a generator that makes really realistic handwritten images. GANs have been applied a few times in population genetics. In this top example, they used GANs to try and create artificial genomes. A similar approach that I'm not showing here tried to use GANs to create artificial alignments as well. And here on the bottom is an approach where they used GANs to try and infer parameters of demographic models. And this motivated our work. We wanted to try to use GANs to infer tree topologies. And so to do this, we have to make a couple of changes to a traditional GAN. So we replaced the generator with an evolutionary model that includes a tree topology and branch lengths, and an evolutionary simulator. Here we used OliSim. Using this new generator, we can make fake alignments, and we can then compare these alignments to observed alignments using a discriminator, which is just a convolutional neural network. At each iteration, our discriminator learns to better distinguish our fake and real alignments, and our generator tries to learn to make more realistic alignments, and it actually does this by updating this tree topology. And so we're going to use nearest neighbor interchange moves and subtree pruning and regrafting moves, which are often used in heuristic explorations of tree space, to propose new trees. And when we get trees that make it harder for the discriminator to distinguish fake and real data, we'll accept those trees. And so ideally, by the end of training, we'll have a generator that produces alignments that are similar to our observed alignments. And we can infer that the tree that was used to generate those alignments is the best tree. And this is nice because it allows us to heuristically explore tree space, where we're starting with some tree, we're proposing new trees, and if those trees make it harder for the discriminator to distinguish our simulated alignments from our real alignments, then we can accept those and move through tree space, hopefully to the best tree. And so this is really nice because it gives us a lot of flexibility. Since we can simulate data under many processes, we can consider lots of different processes, because if we can simulate data under it, in theory, we can incorporate it here. And as I mentioned, it gives us this potential to heuristically explore tree space. Okay, so in phyloGAN, our input is a concatenated alignment. We also require some priors. It's not much here. We need things like a uniform prior on a speciation rate so that we can infer branch lengths. And then there are a couple of parameters like the number of iterations you want to use to train the GAN. At the end of training, phyloGAN will output a final species tree. It will also output a distribution of the trees that were visited by the GAN and some performance metrics. phyloGAN has 3 stages. In the first stage, it's going to try and infer a reasonable value for lambda, the speciation rates, so that we're using somewhat reasonable branch lengths throughout training. In the next stage, we're going to start on our topology inference, We'll begin with what's called a pre-training phase, which just gives our discriminator an overall view of the data. And then we have our actual training phase where we're trying to move heuristically through tree space. Okay, so to start, we tried this on a 5-taxon tree. On the y-axis here, I'm showing you the Robinson-Folds distance from the true species tree. And on the x-axis, I'm showing you the sequence length, so the number of base pairs we used. With as few as 5,000 base pairs, we can do fairly well. However, you will see that we do make occasional mistakes. Luckily, it's fairly easy to identify those cases by looking at our training metrics. So here, I'm showing you the loss for the generator and the discriminator. On the top, we see a run where we inferred the correct tree. We can see an early decrease in the generator loss, and we can see a relatively low discriminator loss throughout. But importantly, we do see cases where we move to new trees. On the bottom, I'm showing you an example of a run that didn't work well. Here, the GAN just got stuck and stopped accepting new trees. And so this is what tended to happen when we didn't infer the correct tree. Of course, 5 taxa is not that much more exciting than 4 taxa, so we also tried this on some larger trees. And we found that we had very high accuracy. Again, this is the Robinson-Folds distance from the true species tree on the y-axis. And so we do quite well for up to 10 taxa, but things start to get a bit challenging when we get to 15 taxa. Okay, so I told you that the reason we should use machine learning in phylogenetics is so that we could incorporate processes that lead to heterogeneity. But so far, I've only showed you results under a fairly simple model. And so next, we wanted to try and incorporate incomplete lineage sorting into phyloGAN. So we developed another version of phyloGAN called phyloGAN ILS. And the major change here is to our generator. Now we're going to generate gene trees under a multi-species coalescent model using DendroPy before generating our sequences. Otherwise, we're doing the same thing. We're comparing simulated alignments to real alignments using a discriminator and proposing new species trees using nearest neighbor interchange and subtree pruning and regrafting moves. For ILS, we ran this on a 6-taxon species tree. We used 1,000 training iterations and generated 5,000 gene trees with sequences ranging from 300 to 1,200 base pairs. And we found that in this relatively simple case, we had very high accuracy. So in all of our replicates, phyloGAN accurately inferred the species tree. However, this gets really computationally intensive, so we didn't explore larger trees with this version of phyloGAN. So phyloGAN does extend machine learning approaches beyond quartet trees, and we can incorporate additional processes like ILS into phyloGAN. It's really flexible, so there's potential to incorporate other processes. However, I think the main negative here is that GANs can be really challenging to train. It's really hard to get them to converge on the correct solution sometimes, particularly as trees get larger. So when we got to 15 taxa and as we add more processes. And these can be very computationally intensive. So this takes several hours even to run on a relatively small number of taxa. And so I think those are the major limitations to this moving forward, and exploring different ways of summarizing the data for input into the GANs and ways to improve the training are potential paths forward. My lab is also thinking about other ways we can use machine learning in phylogenetics outside of inferring tree topologies. These are some very preliminary results. But for example, we're interested in inferring rates of duplication and loss while accounting for processes like incomplete lineage sorting. And for this, we've tried graph neural networks. And these are really nice because they can take gene trees directly as input, as well as fully connected neural networks that analyze summary statistics calculated from gene trees. And we see some really promising results. So here, I'm showing you the actual rate of gene duplication versus the rate predicted by our model. And we can see that both our fully connected neural network and our graph neural network have fairly high accuracy across this parameter space. So we're excited about continuing to explore this moving forward. And in general, I think inferring tree topologies is a really hard problem to address with machine learning, but there are lots of promising solutions that are being proposed. But it's important to think about other ways that we can use these approaches in phylogenetics to infer parameters that we're interested in. Okay, so now I'm going to shift gears a little bit, and I'm going to start talking more about population genetic inference and specifically demographic model selection. In demographic inference, we're trying to identify processes that shape genetic variation in our focal systems. So for example, we might want to infer divergences between populations. We might want to infer population size changes. And we might want to infer things like gene flow between populations. This has lots of applications in evolutionary biology. It can help us understand the importance of different biotic and abiotic factors in driving diversification in our focal system. It can help us to investigate the mode of speciation. So for example, when people infer gene flow during speciation, that indicates that speciation happened in the presence of gene flow, whereas if we infer an absence of gene flow, that might indicate that speciation happened due to geographic isolation. We can also use demographic model selection to do things like delimit species and to identify null models for detecting selection. However, the massive model space, again, is going to make this a challenging endeavor. So I talked about the massive model space that we see in phylogenetics due to changes in the topology. In demographic model selection, we have to think about changes in the relationships among populations or the topology, as well as several other things. So we might want to compare models with different numbers of populations. We might want to compare models that differ in the presence or absence of population size changes in individual populations. And we probably want to compare models that differ in the presence and timing of gene flow between populations. And so this leads to a really large model space with lots of really complicated models. And comparing large numbers of complex models can be intractable under standard likelihood and Bayesian frameworks. This has led to a growing popularity of likelihood-free methods that rely on simulations to compare models. These include approximate likelihood methods like FastMCOL2, approximate Bayesian computation, and supervised machine learning. There are lots of current approaches for doing this, but they tend to be limited in two major ways. So first is in the data that we use for inference. A lot of approaches rely only on the site frequency spectrum or some set of summary statistics. The second is in the models that are available for comparison. I think that the flexibility of supervised machine learning makes it an ideal approach for addressing some of these limitations. As I mentioned before, we can compare any models under which we can simulate data, so that's great. And machine learning gives us a lot of flexibility in terms of the data that we can use for inference. However, currently machine learning approaches for doing this tend to be either tailored to specific systems, limited in the data types they can consider, or both. And so my lab's been working on a Python package called PyPy. PyPy is a package for demographic model selection, and our goals in developing this are really to develop a user-friendly pipeline so that empiricists working in all kinds of systems can apply these supervised machine learnings to demographic model selection. We want to increase the flexibility in the data types considered, and we want to expand the breadth of models that can be considered. So the first step in this is to generate a set of models that the user wants to compare. We do this using a few different approaches. So one way is we can automatically generate models. We ask the user for a species tree or a set of species trees, some information about which populations should be able to potentially experience migration, and some priors on things like divergence times and mutation rates. Then PyPy will output some models. These models can vary in the number of populations. They can vary in the relationships among populations and in the presence or absence of migration. So in this example, PyPy has output 4 different models. We have a model where all of our populations are collapsed. We have a 2-population model. We have a 3-population model. And we have a model with gene flow between 2 populations. Alternatively, the user can specify their own models in simple text files. Here we can implement processes like divergences, continuous gene flow, admixture events, population size changes, and population growth rate changes. And we're working on implementing variation in recombination and mutation rates across the genome. We're also implementing variation in effective population sizes, which can be used to model to kind of mimic the effects of background selection, and ghost integration. And then finally, we're making it where it's easy to import external simulations into PopEye. Particularly, we want users to be able to import tree sequences that are generated in SLIM, which is a program for forward-in-time simulations. And this allows users to include selection and spatially explicit processes in their model set. Next, PopEye will simulate data. We simulate data using MS Prime, and PopEye does a lot of things behind the scenes to generate simulated data that's similar to your empirical data with respect to things like the distribution of locus lengths, the number of loci, and the number of SNPs per locus. Finally, we'll encode the data. We encode the data as multidimensional site frequency spectra or joint site frequency spectra. If you're not familiar with site frequency spectra, these are encodings of SNP data, where essentially this is a site frequency spectrum for two populations. And this cell here, for example, counts the number of times that the derived allele occurs once in this population and 0 times in this population. And so this is a really nice summary of SNP data. We can also encode SNP alignments directly for input into PopEye. And we're working on encoding gene trees, several population genetic summary statistics, and some other ways for formatting alignments. After this, we're ready to train our machine learning algorithms. And right now we implement random forests, fully connected neural networks, and convolutional neural networks. The convolutional neural networks are probably the most powerful thing currently implemented. These can work on our site frequency spectra, treating those as images, which I think is a really convenient way to analyze site frequency spectra. And they can also analyze the SNP alignments directly. We're currently implementing graph neural networks for analyzing gene trees and networks that can take multiple data formats as input so that users could use both site frequency spectra and gene trees to make inferences. Okay, so this is just an overview of the PopEye workflow. We start by generating models, we simulate data, encode data, and then train algorithms. And these algorithms are then ready to be applied to your empirical system. If you're interested in checking this out, it's available on our lab GitHub. We've done some simulation studies to assess how well PopEye works. So I'm just going to show you results from this simple simulation study here. Here we started with 4 models. We have a 3-population model, and we have the presence or absence of gene flow between pairs of populations. First, we trained a convolutional neural network to analyze our site frequency spectra. And so the input into this neural network is joint site frequency spectra for each pair of populations in the model. And we found that we had really high accuracy. So on the y-axis here, we have the true class, and on the x-axis, we have the predicted class. And so we almost always, I think in this case, we had an almost 0% error rate. We had a couple of errors between models that differ only in the presence of gene flow between closely related populations. To the right here, I have the results from a convolutional neural network that analyzed the SNP alignment directly as an image. Again, we had low overall error rates, so our overall error rate was around 5%, but we do see more mistakes between models that differ only in whether there's gene flow between these sister populations in this case. And so I think these results are really promising, and I'm hoping that this is going to be a flexible framework that's really useful for a lot of people. We're working on, as I mentioned, incorporating more processes, including ghost integration, variation and recombination and mutation rates, and selection. We're also incorporating more summaries of our data, including gene trees. And then finally, what I'm going to spend the rest of the talk talking about is the use of machine learning approaches to deal with model misspecification. Okay, so I've been talking a lot about supervised machine learning, And you've seen this figure before. And what I'm highlighting here is just that we have to use simulations under some models to make inference. All models are necessarily going to be a simplification of what's really happening in your empirical system. And so we're really interested in what happens when the simulations that we're using to train machine learning algorithms are not very realistic. So for example, let's imagine that we train our machine learning algorithm to detect integration between a pair of populations. So for training, we simulate lots of data sets without integration and lots of data sets with integration. However, in our empirical system, there's actually been integration from some third unsampled population into one of our two focal populations. And so this would have been a more realistic model, but for whatever reason, we didn't include this in training, perhaps because we didn't sample this third population and we may not have known that it existed. In machine learning, this is called a domain shift. A domain shift occurs when the training data and the target data arise from different distributions. So the canonical example in machine learning comes from digit classification. If you've done much machine learning, you've probably run into this data set of handwritten digits. This is really commonly used to develop machine learning algorithms. And so let's imagine that someone built a classifier to identify digits from these handwritten digits. This is the source data or the training data. If they then apply that classifier to a data set of street house signs and try to identify digits in the street house signs, that is an example of a domain shift because the target data comes from a different distribution than the source data. And it's known that then this often decreases the accuracy of machine learning algorithms. In population genetics, this could look like what I just mentioned, where we trained an algorithm under two population models, but actually there's been integration from a third unsampled population into one of our two populations. Before I talk more about this work, I just wanted to say that this work was led by a postdoc in my lab, Carrie Cobb. And Carrie and I focused on the problem of trying to detect integration between pairs of population, which is a central goal in many studies of evolutionary biology. Lots of people want to do this, so many methods exist to do this. I've mentioned some of these before today. So we have the methods based on the site frequency spectrum, which include things like FastSimCoal2, and methods like BPP, which uses a multi-species coalescent with integration to analyze sequence data. And these methods are really powerful, but these methods, along with the supervised machine learning methods that we've been talking about today, rely on models. And these models make assumptions about the processes that shape genetic variation. And so, for example, most of the methods that we talked about are going to ignore the potential of integration from unsampled populations, also called ghost integration. And so perhaps there's been integration from an extinct population or a population that just wasn't sampled. Most of these models are also going to ignore selection, and they're going to ignore things like sequencing error and many other things. This might be okay. All models are going to be simplifications, so we have to ignore some processes. However, we know that violations of some of these assumptions can mislead tests for integration. So for example, ignoring ghost integration will lead to false inferences of integration using many different approaches. And ignoring selection can also lead to false inferences of integration, particularly for site frequency spectrum-based methods. So we wanted to ask how simulation misspecifications affect machine learning approaches for inferring integration. Since these approaches are still relying on models to generate their training data, we expected that there would be some negative impact. To test this, first we just built a convolutional neural network to detect integration. And so in this case, we simulated training data with or without integration between our two focal populations. Then we summarized our data as a site frequency spectrum, and we used a convolutional neural network to extract information from that site frequency spectrum. The features that we extracted were then used as input into a fully connected neural network. which then predicted the probability of each of our two models, a model without introgression and a model with introgression. So first, we applied this to data simulated under these same models. And we found that this network was highly accurate on testing data simulated under the same models as training data. So if we simulate data with introgression, we always detect introgression. And if we simulate data without introgression, we never detect introgression. So that's great. But we're interested in the case where processes that generated the data we want to classify, also called the target data, perhaps empirical data, were not included in our training simulations. And so next, we applied our network to data that was generated with introgression from an unsampled population into one of our two local populations. In this case, we found that we did have lower accuracy. So our goal is only to detect introgression between these focal populations. When introgression did occur between these populations, we usually still detect it, but we do see some false negatives. And when introgression does not occur between these focal taxa, we get a relatively high rate of false positives. We can also visualize this as an ROC curve. Here on the y-axis, I'm showing the true positive rate, which is ideally close to 1. And on the x-axis, I'm showing the false positive rate, which is ideally 0. And in a perfect classifier, we would see something that looks like the blue line here, where we go straight up to 1. And that's what happens when we have no model misspecification, because our classifier doesn't make any errors. In the presence of our misspecification, which was ghost introgression, we see lower accuracy, and so we see these orange curves. We have 10 different replicates. We also have 10 replicates without a misspecification, but you just can't see them here. Okay, so we've identified a problem, but what can we do about it? We wanted to try an approach called domain adaptation. In domain adaptation, we try to train networks that perform well, even when we have a shift between our source and target data or a domain shift. So in this case, we still have our source or training data. These are data sets where we know whether or not introgression happened between the focal population, but we also have our target data. And one thing I really want to emphasize here is that we don't need any information about our target data. We have no idea whether or not introgression happened between our focal populations in our target data set. So these can be empirical data. We still have a convolutional neural network that we use to extract information from a site frequency spectrum. Those features will still be fed into a fully connected neural network for classification, which will aim to maximize our ability to distinguish between the models, a model with or without introgression between the focal populations. But we're adding a new layer here, and this is called our discrimination network. This network is going to take those same features as input, but it's going to try and distinguish whether data sets came from the source distribution or the target distribution. And what we're going to do is we're going to try to extract features that minimize our ability to distinguish amongst those distributions. And so hopefully by the end of training, we have a feature extractor that can extract information that works really well for distinguishing between our two focal models, but cannot be used to distinguish between our source data and our target data. Okay, so we tried this with ghost introgression. On the left here, I'm showing you the previous results, where the orange lines show you the classifiers applied to data with ghost introgression. This is just the original classifier. On the right, I'm showing you the results from our domain adaptation network. And so here we see that even when we have a model misspecification, we have really high accuracy. We have a nearly perfect classifier, even in the presence of ghost introgression. And so I think this is really exciting because we didn't need to know anything about our target data sets, but we could correct for this issue that was caused by the presence of ghost introgression. Of course, all these results were on simulated data, which is always cleaner, and we know exactly what our model violation was. So we also wanted to try and apply this to empirical data. For this, we focused on data from brown bears. So a lot of previous work has shown that introgressions between the brown bears from the ABC Islands, shown here in green, and polar bears will mislead tests for introgression between populations of brown bears. And so we took whole genomes from lots of different brown bear populations, and we used our neural networks to test for introgression between our ABC Island bears and these other brown bear populations. Here, I'm just showing you the results without domain adaptation. In this top graph, I'm showing you the probability of introgression. And so yellow means there's a higher probability of introgression. Each column here is a different chromosome. We analyze the side frequency spectra from each chromosome separately. And what we see is lots of support for introgression. And I'm highlighting here in red populations that are geographically somewhat isolated from these ABC Island bears. And we see lots of support for introgression between these geographically isolated populations, which we might think is not particularly realistic. On the bottom, I'm showing you the results from our threshold classifier. In this case, we only classified something as introgressed if the probability of introgression was above 0.95. Even with this conservative threshold, we see lots of support for introgression. Next, we applied domain adaptation. On the top, you can see that we have lower probabilities of migration overall. And on the bottom, when we use our thresholding approach, we now only have a single test showing support for introgression between the ABC Island bears and a geographically distant population. Of course, these are empirical data, so it's hard to know what the true answer is, and it's hard to know exactly what model violations we had. But I think that our results with domain adaptation are more reliable than our results without domain adaptation in this case. So violations of the models that we use to simulate training data can lead to high error rates when we use machine learning methods to do things like detect introgression and presumably for other tasks in demographic model selection. But domain adaptation can lead to accurate inferences, and it doesn't require that we have any knowledge of our target domain, which can just be our empirical data. Our initial application of this approach to empirical data from brown bears suggests that it may reduce false positives potentially caused by ghost introgression. We're really excited to continue working on this moving forward. So one really nice thing about this approach is that it's agnostic to the specific model violation. And so we could apply it to a number of other model violations that plague population genetic inferences, like background selection, and we're looking forward to trying that. In this case, we use networks that analyze site frequency spectra, but it's also possible for us to use networks that analyze alignments directly, and this may increase our power for some more complicated models. And then finally, we are working on incorporating this into Popeye. Overall, I think machine learning approaches are really powerful when we want to analyze large genomic data sets using complicated models. And these approaches have been useful in both phylogenetics and for demographic model selection. With any approach, there are going to be negatives, and I think that simulation misspecification is a major pitfall to these supervised machine learning approaches, but I'm really excited about domain adaptation as a potential path forward. With that, I'd like to thank my lab at Mississippi State, including Carrie, the postdoc who worked on a lot of this domain adaptation stuff. I'd like to thank Matt Hahn, who was my postdoc advisor when I was working on phyloGAN, and also phyloGAN was funded in part by a fellowship through the National Science Foundation. And with that, I should have plenty of time for questions.
Thank you very much, Mike, and that was awesome. Well, as I said, feel free to type in your questions if you don't want to unmute, but if you do want to unmute, raise your hand and you can do that. But for now, let me just read. There are a couple of questions in the Q&A. Let me read them to you. So first is Oliver Leon asking, how does phylogen compare with traditional machine learning methods for tree search in terms of time? How long did it take to infer 6 taxa topologies?
It's not very fast. So I think for 6 taxa, it took somewhere around 3 or 4 hours. So it's quite slow. That's one of the main limitations, I think, of using that kind of approach. So it can work for larger numbers of taxa than traditional machine learning approaches, but it's certainly much slower than other phylogenetic methods.
And then David Horwath is asking, what would be the best AI method, pop AI question, to identify the best trees from within species datasets? Say between several hundred members of a diversity panel for a crop plant like canola. Also, how does it deal with polyploids that are common in plant species?
Yeah, it's a good question. So I think the first question is just if you just wanted to compare topologies. I mean, I think you said like hundreds of individuals.
Within species, yeah.
I mean, if you want to compare topologies when you have hundreds of individuals, you either would need to assign those to populations first, or probably use a different approach. But if you can assign those to populations, and then you're just trying to compare models that have different relationships among populations, then I think so far we've gotten the best results from the convolutional neural networks on joint site frequency spectra, because you have information about how closely related each pair of populations is there. But one nice thing is that you get the output of all the models that you train really quickly, so you can easily compare which summaries of the data and network methods work best. But I suspect that those networks might perform best. And I've forgotten the second part of the question, sorry.
Oh yeah, the polyploids. Is there a way to deal with that?
We can deal with that. It wouldn't deal with it right now, right off the bat, unless you had phase data. But typically when we're simulating data for these types of methods, we're simulating chromosomes. So you would just simulate more chromosomes, right? So for a diploid individual, we actually simulate 2 chromosomes. So for a tetraploid individual, we would just need to simulate 4 chromosomes.
I had kind of a related question. You in the first stage of POPA, you mentioned that you can suggest the topologies, the population topologies. Is there like what's the way that you evaluate them, or you just enumerate all of them?
We will enumerate all of the topologies that the user provides. So they can provide like a list of species trees, and we'll enumerate all of those. There's not really a great way for us to cut down on them except using like prior information that the user has.
Yes, makes sense. Valeria Vasilieva is asking, in phylogen, you limit the number of variable sites to a specific number. And if there is less than this number, you impute missing values with a constant. May I ask, why do you fix on a specific number of variable sites?
Right, so we don't impute missing data. We just we have a missing data value that we just put into the matrix. But the reason that we do that is because convolutional neural networks, designing CNNs that are actually able to analyze data sets that have different numbers of columns is really hard. So you can do it. And there's a really nice paper that came out in Molecular Ecology in 2021 where they did this for inferring population size changes. The problem when you try to do that for networks that you're using to make phylogenetic inferences is that the approaches that you have to use to make a network that can analyze data with different numbers of columns summarize your data in really specific ways. So you have to make filters that of really specific sizes. And what we found, and I think some others have found as well, is that those networks tend to not work very well for phylogenetic inference. And so I think finding a good way to create convolutional neural networks that are able to accommodate varying input sizes in a more natural way than just padding with zeros or something is a really important thing to figure out in phylogenetics and machine learning. But I don't have a good answer.
Would something like bootstrapping work for like standardizing the length of sequences?
Right. So I guess you could just pick you'd have to still pick a minimum. Yeah, you could just pick a minimum length, and if data sets had more than that, just treat them as different images. Is that what you mean?
Yeah, kind of. If you to standardize the
Right.
sequence length, just choose a number that you need and bootstrap it or something. So
Right.
slightly
Yeah.
replacement. Yeah, I'm not
Yeah.
sure. If
Certainly, especially when you're applying it to empirical data, that would be a good approach to do it, to make a network that was trained on a different size data set work.
Edward Braun is asking, could you give a little more detail about the model for phylogen ILS? Were you assuming a molecular clock?
Yes, we were. Yeah, we didn't get more complicated because it was so slow, even in the simple case, that it didn't seem to make sense to pursue adding more complication there.
Makes sense. I was actually also curious. When you're doing phylogen, you mentioned that you're doing NNIs and SPRs to change the tree topology. But are you tweaking the simulation model at all, the substitution model?
No, we didn't try that. So you could do that. Again, I think if we were able to find a way to train it that was more efficient, such that we thought it was going to be more applicable to like real case scenarios, then that might be worth looking into. But since it was already slow, didn't add more complication there either.
Makes sense. Luca Micheli is asking, have you had any experience in using population genetic summary statistics to train a convolutional neural network? How does it compare with using images of SFS?
Yeah, that's a good question. I haven't used population genetics summary statistics to train a convolutional neural network, but I have used them to train fully connected neural networks or random forests. In general, it's just going to depend on how much information your statistics capture. Often, for some types of models, using summary statistics works really well and gives you really high power. So sometimes they work really well. If you get really complicated models, then sometimes you might be sacrificing too much information if you have a relatively small number of summary statistics, but often they're really powerful.
Makes sense. If folks have any more questions, please do ask. I had a well, not so quick one, but could you explain quickly a little bit more about the domain adaptation technique? So you mentioned that you're trying to identify features that distinguish models well, but then also features that are those the same features that try to both distinguish and
Yeah, no, that's a good question. So we have this feature extractor, and the features that it extracts, the same features are going to be fed into both of these two networks. And so what's going to happen is this discriminator network is going to force it to extract features that don't differ between these two models. And ideally, if we're using features that don't differ between our source and target domain to distinguish amongst our models, we shouldn't be biased by whatever's happening in our target domain that's not happening in the source domain. So that's kind of the thought process behind it, is that if you can force your network to extract features that can work well for classification, but don't differ across domains, then hopefully you'll be more robust to the differences between these domains. Does that make sense?
and then also the features that you use then for analysis, right?
Sorry, what was the question?
And then those are the features that you use for inference, right? The ones that you extracted, the ones that you
Yes, yep. Yeah, those are what's being used for inference by this classification network.